Good afternoon, and welcome to the Brain and Behavior Research Foundation's Meet the Scientists monthly webinar series. I'm Dr. Jeff Borenstein, President and CEO of the Foundation, and your host for today's webinar. Today, Dr. John Krystal will present Ketamine, Why Now, How, Where Do We Go From Here? The Brain and Behavior Research Foundation is committed to alleviating the suffering caused by mental illness by awarding grants that will lead to advances and breakthroughs in scientific research. The Foundation is the largest private funder of mental health research grants. Since 1987, the Foundation has awarded more than $380 million to fund more than 5,500 grants to more than 4,500 scientists around the world. 100% of all donor contributions for research are invested in our grants to scientists who are working to find breakthroughs in disorders such as addiction, ADHD, anxiety, autism, bipolar disorder, borderline personality disorder, depression, eating disorders, OCD, Ptsd, and schizophrenia. I'm delighted to introduce Dr. John Krystal. Dr. Krystal is chair of the Department of Psychiatry at Yale University School of Medicine. He is a member of our scientific council and was a Foundation Distinguished Investigator Grantee in 2006 as well as 2000 and was an Independent Investigator Grantee in 1997. Today's webinar will begin with Dr. Krystal's presentation. This will be followed by a question and answer period. To submit your questions, please use the questions tab on the control panel on your screen. Please feel free to submit your questions throughout the presentation. Following the presentation, I will ask as many of the questions that we have time to ask. And now, I'm pleased to introduce Dr. Krystal. John, the floor is yours.
Jeff, thank you so much. And, I just want to highlight for everybody on the webinar that BBRF and NARSAD have been incredibly important for the field. They supported some of my own work and so many young faculty members getting their starts in research. And so it's a thrill to be able to be here with you today. Today, I'm going to be talking about ketamine, which is an exciting area. And I'm going to be talking about its application to the treatment of post-traumatic stress disorder, but I'll also cover aspects of how it's being used in the treatment of depression and some related conditions. Now let's see. Okay. I want to acknowledge that the work that I'm going to share with you today has been supported by a variety of different organizations, such as the Department of Veterans Affairs, the National Institute of Mental Health, BBRF, and others. And that the work that I'm going to be talking about is from one of the most important organizations in studying post-traumatic stress disorder, which is the Department of Veterans Affairs National Center for PTSD, which is across many different sites, and our site studies the clinical neuroscience of post-traumatic stress disorder. I also want to acknowledge that I have patents that have been licensed by Janssen Pharmaceutical, Biohaven Pharmaceutical Sciences, related to the treatment of depression, and that I consult to companies mostly about the neurobiology and treatment of schizophrenia. So, where do we begin? Post-traumatic stress disorder is one of the most common psychological casualties of experience in war. It's associated with traumatic events in civilian life, sexual assault, even acute medical illnesses. And yet it's an illness that has been struggling to develop effective medication treatments. My colleagues and I joined together recently to both call attention to the need for advances in this area and to highlight that there are some new opportunities to take advantage of in moving forward in post-traumatic stress disorder. The standard treatment medication treatment for post-traumatic stress disorder are the class of medications known as SSRIs. These are the medications like Prozac, like Zoloft, and so on. These medications are helpful for many people, but overall, the efficacy is limited. In other words, about a 10% reduction in symptoms relative to people treated with placebo. And the effect size, in other words, how much benefit can people expect, overall is a little bit smaller from the antidepressants than from the evidence-based psychotherapies for PTSD, such as cognitive processing therapy or progressive exposure therapy. And despite years of research, we still do not know whether being on medication actually helps the psychotherapies to work more effectively or not. These medications take a long time to kick in, if you will. So the average time for until sertraline can be shown to be more effective than placebo is after 10 weeks of treatment. And some groups of people with PTSD, such as men who've been in the military, combat veterans, tend to have poorer outcomes with the SSRI medications than do the more acute or single episode trauma populations. There is some further discouraging news, which is that there have been a number of large-scale studies funded by the Department of Veterans Affairs, two medication studies, one that I led on risperidone, and one led by my colleague, Dr. Murray Raskin, studying prazosin, both of which were promising treatments based on initial studies, but which proved not to be effective in large-scale definitive trials. So my presentation today is really, how do we move forward from here? How do we get through this kind of glass ceiling that seems to be preventing us from discovering new and more effective treatments for PTSD? And the case that I'm going to make is that it has to do with a deeper understanding of the biology of post-traumatic stress disorder and depression, and that this deeper understanding leads us to test medications like ketamine as a treatment for PTSD. And I'll talk a little bit about the implications of this for the future. So the first thing to know about the biology of ketamine and PTSD is that it involves a chemical system in the brain called glutamate. Now, you may be familiar with chemicals, brain chemicals like norepinephrine and serotonin. These are chemicals based in the very primitive parts of the brain. But glutamate is the main information thoroughfare of the cerebral cortex. What do I mean by the main thoroughfare? I'm going to show you now. What you're looking at is a slice through a very small section of the cerebral cortex, and you see all of those colorful cells. These are the main workhorse cells of the cortex, known as pyramidal neurons, and the main chemical messenger, in fact, almost the principal, if not only chemical messenger that this group of cells uses is glutamate. And we're going to zoom in on these glutamate nerve cells, because I want to make a particular point about the nature of communication in the cerebral cortex. Okay, we're focusing in on one glutamate nerve cell, a real nerve cell that's been traced under electron microscopy at ultra-high resolution. And in red, you see all those little blebs sticking out. Those red blebs are called dendritic spines, and they are the site that synaptic connections, in other words, the place where the communication between glutamate nerve cells actually takes place. And they're critically important for the functions of the cortex. You'll notice that there's a big barrel with many different kinds of colors, and we're going to zoom in on that barrel and look at the complexity of the connections. By the way, let me say that these were electron micrographic tracings done by Jeff Lichtman and his colleagues at at Harvard University. Okay, now we're zooming in just that one little segment of that one little neuron, and we're going to blow it up. And you can see that that one little segment of that one little neuron, in order for it to work functionally, has to have hundreds, if not millions, of connections between that nerve cell and the neighboring nerve cells and the other supporting cells of the brain. We're going to look at that in a little bit more detail now, where you can see the axons, which are the outputs, the dendrites, which are the inputs of nerve cells, and the astrocytes, which are the supporting cells. So at this level, you can see why the brain behaves in so many ways like a computer, because there are millions and billions and trillions of synaptic connections in the brain, and those connections are the information thoroughfare. And in the cortex, the vast majority of the connections are excitatory axons, those which release glutamate, and the spiny dendrites, those which receive glutamate inputs. So I'm talking to you today about a stress problem. And for many people, when you think of stress, you think of the most studied stress hormone in the body. That is cortisol. Now, there's a lot of evidence coming up from basic research studies that chronic high levels of cortisol, as you see with major depression, or as you see potentially in post-traumatic stress disorder, can be harmful for the brain. It produces a loss of the synaptic connections in many regions involved in the regulation of emotions. And it causes the glia, or support cells, to be less able to restore the balance of glutamate metabolism in the brain. But cortisol is also helpful. And you need cortisol for the brain to adapt and respond to at some level to reduce inflammation. And it's possible that in PTSD that there is inadequate cortisol, not enough to keep up with the demands. So cortisol can be harmful if it's too much for too long, but it may also be harmful if it's too little for too brief a period. One of the ways that we can look at that is with one of the important glutamate receptors in the brain. This is called the mGlu-R5 glutamate receptor. And when activated, it helps to remodel synapses in the context of experience. In other words, it helps us learn about fear. It also helps us to get over the fear, to extinguish the fear. So we want to have a well-balanced signaling through the mGlu-R5 receptor. There's a possibility that one of the ways that glucocorticoids contribute to stress vulnerability is by altering the function of the mGlu-R5 receptor. Acute stress causes the mGlu-R5 receptor to be decreased, whereas chronic and repeated stress causes the mGlu-R5 receptor to be increased. So that we see in major depressive disorder, generally speaking, a reduction in mGlu-R5, less mGlu-R5, more severe depression. But what about post-traumatic stress disorder? Is it like acute stress, or is it like chronic stress? We recently completed a study involving PET scans in people with PTSD, and we found robust and widespread increases in the number of mGlu-R5 receptors in PTSD. In other words, the pattern associated with chronic repeated stress. And the extent to which there was an increase in the mGlu-R5 receptor correlated with the severity of PTSD symptoms. More mGlu-R5, more PTSD symptoms. Now, in order to understand why you get that, you need to understand that DNA is converted, expresses, and makes mRNA, which is called messenger RNA. And messenger RNA is the basis for a protein. And the protein that we're talking about now is the mGluR5 receptor. What I'm showing you here is data from one of the first brain bank analyses of postmortem brain tissue ever conducted in PTSD. And what this shows is that the mGluR5 receptor is not increased in PTSD, but a related protein, a related transcript, mRNA levels for something called SHANK1, which I'll tell you about in just a minute, is increased. SHANK1 serves to anchor the mGluR5 receptor to the cell membrane. So normally we just have maybe a certain amount of mGluR5 receptors. If you had an increase in SHANK1, that would help to get more mGluR5 receptors. So mGluR5 upregulation in PTSD points to one role for glutamate signaling in PTSD, and that there may be treatments that normalize that, perhaps by blocking the glucocorticoid receptors or blocking the mGluR5 receptors. And there are a class of drugs that we're now testing called mGluR5 negative modulators or blockers, and ketamine. I bet you were wondering when I was going to use that word again. One of the reasons that cortisol and glutamate are so closely linked, as I mentioned before, is that glutamate also causes a loss of dendritic spines. On the normal state, on the upper left, in the depressed state, in the middle. And if we use regular antidepressant treatments and treat people with depression for months and months, we can see increases in connections in the brain, or in animals. If you give chronic administration of antidepressants, you can see regrowth of dendritic spines, which suggests a hypothesis about connections in PTSD, that if there are stress-induced loss of synaptic connections, that you get cognitive functions, and that you have difficulty in neuroplasticity. By that I mean that your brain has trouble remodeling the synaptic connections in order to alleviate the trauma-related symptoms. And that we might be able to treat some PTSD symptoms by restoring the connections that have been lost by stress and enhancing the capacity to respond to treatment. Now, there's a lot of evidence that suggests that there may be a reduction in synaptic connections in the brain. The first MRI study in PTSD was conducted in our center and published in 1995, which suggested that an area in the brain called the hippocampus was reduced the volume of that area was reduced in PTSD. Nowadays, using functional MRI, we can show that functional connections in the brain are reduced in PTSD in relation or correlated with the overall severity of PTSD and symptoms such as numbing and hyperarousal. Standard for treatments for PTSD, like paroxetine or Paxil, can reduce symptoms of PTSD, and for long-term treatment, 6 months of treatment, may even increase the volume of some of the areas that are reduced in PTSD. In other words, the volume of the hippocampus, this area involved in memory, increases. So I've given you some reason to believe that there's reduction in synaptic connections in the brain as a result of traumatic stress, and that PTSD symptoms are associated with MRI changes, and that long-term antidepressant treatment can improve connections. But what if we could do this better and quicker? And that's where ketamine comes in. Ketamine has been called by Time magazine the anti-antidepressant because it doesn't work like any other treatment that we have had for the treatment of depression. So how did we discover ketamine? Well, we started by shifting our focus. We tried understanding the biology of depression. My colleague, Dennis Charney, led a series of studies showing that depleting the body of serotonin or depleting the body of norepinephrine or depleting the body of norepinephrine and serotonin did not produce depression in people. This suggested to us that maybe the biology of depression and stress-related disorders didn't primarily live in the primitive parts of the brain that used serotonin or norepinephrine to communicate, but rather in the higher centers of the brain, the cerebral cortex, where glutamate is the predominant neurotransmitter. To test that idea, we gave a single dose of the drug called ketamine to treat patients with depression who mostly had very resistant symptoms of depression. And what you see in the top was the most striking finding of the study, which is that a single dose of ketamine within 24 hours produced a predominant clinical response in the patients that we studied. Some people completely remitted from their depression. They were all better, not just a little better, but basically their depression had melted away within 24 hours and stayed away for several days afterwards. For some people, 2 weeks or more after a single dose. And what's striking and important to understand about ketamine, and often misunderstood, is that the effects of ketamine, the behavioral effects of ketamine, are completely gone by 2 hours. So what we see as the clinical benefit of ketamine is not how people subjectively feel while ketamine is in their body, but rather the body and brain's reaction to having been exposed to ketamine. There's no ketamine in the body at the time when people are reporting the greatest clinical benefit from ketamine. So you're not high, you're not intoxicated, you're not feeling any drug effect except the reaction to the drug that you received the day or several days before. Now, one of the things about ketamine that's important to understand is that it has obvious effects. It transiently produces euphoria. It transiently produces perceptual changes. And therefore, people wanted to compare it rather than to an inert placebo like salt water, to a drug which itself had strong behavioral effects, a drug called midazolam, which is like Valium. And here on the left-hand side of the page, in a study by Sanjay Matthew and James Murrow, you see that ketamine not only produces stronger antidepressant effects than midazolam, but on the right-hand side, you can see that these antidepressant effects from a single dose of ketamine last for days and days, while the benefits from midazolam are pretty much gone by day 2. An important area of study related to ketamine is its reported effectiveness in reducing suicidal ideation. And there are now many studies related to further understanding that phenomena. And here is one study which shows on the left-hand side that midazolam does not produce much benefit on suicidal ideation, but ketamine robustly reduces suicidal ideation. And if you fail to respond to midazolam and were randomized then, or were able to get ketamine, that's the orange group, you can see that even the non-responders to midazolam do very well in terms of reductions in suicidal ideation while on ketamine. And those who respond to ketamine sustain their improvement over 6 weeks. There are other drugs that are being developed to try to take advantage of the breakthroughs with ketamine. The drug that's farthest along is called S-ketamine, and it's licensed by Johnson & Johnson or Janssen Pharmaceuticals. And they have pretty much completed their phase 3 studies and are getting ready to go to the FDA to seek a drug approval sometime in the remainder of the year. Like ketamine, S-ketamine, and I should add that Janssen licensed our patent for ketamine. So, like regular ketamine, S-ketamine, which contains just the S version of ketamine. Ketamine comes in two flavors, an R-ketamine and an S-ketamine, that are mirror images of each other. The S-ketamine is more potent and selective for NMDA receptors, generally speaking, and it produces dose-related antidepressant effects with single doses. And most striking to me is this study showing how S-ketamine prevents relapse. This was a study conducted by Janssen in which they evaluated people who responded to the combination of an antidepressant medication and S-ketamine. And then when they responded, so they were better, they randomized people to staying on S-ketamine or getting a placebo instead of ketamine. And even though people were still on what had previously been their usual antidepressant, over half of the patients on antidepressant plus placebo relapsed over about 9 months. The relapse rate on people who stayed on ketamine was 25.8%, or about half, less than half, of the relapse rate on people who treated with antidepressant with placebo. So not only is ketamine rapidly active, it's also effective in delaying or preventing relapse. We can Janssen has studied this on an even longer scale, out to 48 weeks, and in 600 patients, showing this sustained benefit. And this slide shows that ketamine, when used in this way, S-ketamine, is very well tolerated. So, in this study, 20 patients stopped taking their oral antidepressants, even though these were medications, actually a total of 33 patients, stopped taking their oral antidepressants, even though these were medications they had taken for a very long time. The number of people who stopped taking ketamine was 76, but ketamine was a new medication, one that had been added relatively recently, and this very high retention rate is indicative of the high degree of tolerability of ketamine. The kinds of side effects that people got, the most common ones were common symptoms of depression, such as anxiety and depression and suicidal ideation, or common effects of ketamine, such as dizziness, perceptual alterations, nausea or vomiting. Ketamine has now undergone wide and intensive study. It appears to be very safe. The dissociative or perceptual symptoms of ketamine that occur transiently with administration are very manageable in routine settings. Nausea can be managed well. If necessary, we can give ondansetron, which manages that symptoms. And while ketamine can be a drug of abuse, we have not had cases of drug abuse when ketamine is administered only in the clinic. It seems to be effective in treatment-resistant depression. Clinic reviews suggest that the response rates are around 75%, which is an incredibly high response rate. There may be evidence, very early evidence of synergy with psychotherapies like CBT. It seems to be effective even in people who fail to respond to electroconvulsive therapy, and in a wide group of people who generally don't respond to antidepressants very well. People with bipolar disorder, people with psychotic depression, people with high anxiety or agitation, or patients with comorbid pain. And as I've shown you, generally with biweekly or even monthly administration. In other words, once a month. About 40% of our patients in our ketamine clinic get infusions of ketamine now once a month or even less frequently. We can sustain the benefits of ketamine. Ketamine is now being used in other settings because it's good for pain and good for depression. Scott Irwin is testing it in hospice patients and other chronic pain populations. Dennis Charney did the first study of ketamine for PTSD. And in the PTSD patients, what you see is that it not only alleviates the symptoms of PTSD, and it alleviates the symptoms of depression in those patients. Depression is a very common symptom of PTSD, and one that surprisingly often does not respond very well to traditional antidepressant medications. In the patients, in the analysis of the PTSD symptoms, the effects of ketamine were still beneficial if you excluded the patients who had depression as a comorbid symptom, or if you statistically adjusted for the severity of depression prior to ketamine infusion. So this appears to be a direct effect on PTSD symptoms. And there are now a growing series of replications supporting the effectiveness of ketamine for PTSD. We are now doing the largest ketamine trial in veterans and active duty military with PTSD. We hope to get 200 patients. We're looking at the dose-related safety and effectiveness over 4 weeks of ketamine treatment and then 4 weeks of post-administration follow-up. We hope to have results in another year and a half or so. So what I've suggested is that ketamine may play a role in rapidly treating antidepressant-resistant symptoms of PTSD, including suicidal ideation, which may help to mitigate or shorten hospitalization and prevent missed work days or days where people can't take care of their families or engage in other productive activities. So where do we go from here? One, we'd like to be able to optimize the use of ketamine for PTSD. And we want to continue the search for alternatives to ketamine that might preserve its effectiveness but increase the utility of ketamine. So one intriguing idea that goes back to the earlier parts of my presentation, I suggested that one of the problems that prevented people with PTSD from getting better from their fears was that the loss of synaptic connections in the brain was reducing plasticity, reducing the capacity to extinguish fear. We now have data from animal models, particularly from the work of Matt Gergenti and Ron Duman's lab in our center, that if we give ketamine, we can produce far more rapid extinction of fear in animals than if we just do the fear extinction by itself. And for us, that's really important because we can show in behavioral and neuroimaging studies problems with the extinction of fear in PTSD. With regards to finding alternatives to ketamine, in order to do that, we need to really understand how ketamine works. And here what I'm showing is some of the early data from Ron Duman's lab that first pointed to this important idea that ketamine might restore synaptic connections. On the left-hand side, you see a dendrite or input to a nerve cell. And you see all the arrows pointing to the dendritic spines that I showed you before. In the middle, you see what happens in animals that undergo chronic stress. You can see that the number of those dendritic spines, and therefore the number of synaptic connections in the brain, goes way down. On the right-hand side, you see what happens 24 hours after a single day a dose of ketamine. The incredible sprouting of those dendritic spines right up to the density that these cells had before stress. So it looks like one of the ways that ketamine works is by helping the brain to regrow those synaptic connections, which are so important for cognition and brain function. So I showed you evidence that that happened in animals. What I'm showing you in this picture is evidence suggestive that that happens in people. And I'm showing you some data first in major depression, where you can see in blue areas of the brain that show reduced functional connectivity. In other words, they're not communicating so effectively with each other, suggestive of an impairment in structural connectivity. 24 hours after a single dose of ketamine, you see that those areas of deficient communication are restored. So it looks like functionally, as well as structurally, a single dose of ketamine has the capacity to regrow synaptic connections and re-enable the parts of the brain involved in the regulation of emotion to function better. Now, I'm going to go through this relatively quickly, but I want to make one point. That you see all these steps in the pathway, and I could list them for you. Blockade of NMDA receptors, reduced stimulation of, reduced inhibition of glutamate nerve cells, increase in glutamate release in the brain, stimulation of AMPA glutamate receptors, elevation of a nerve growth factor in the brain, stimulation of a pathway that leads to the formation of dendritic spines. And all of this happens when we give ketamine. And each of those proteins, each of those steps is a potential new target. We could try to think about how we could activate mTOR. We could think about how we would elevate BDNF. We can think about how we can enhance the stimulation of AMPA receptors and so on. So one discovery, ketamine, is actually the tip of an iceberg for potentially a whole new generation of pharmacologic approaches, drug treatments for mood disorders like depression and PTSD. One of those is our old friend, the mGluR5 receptor. If we give people, healthy people, a single dose of ketamine, you see that the brightness in the brain goes down in the third row. The reduction in brightness means that ketamine is causing a reduction in mGluR5 receptor availability. And when we do the same thing in depressed patients, the ketamine stimulation of the reduction in mGluR5 receptors correlates with improvement in depression. So we have many themes, many new directions. This tells us that we might be able to treat depression by blocking mGluR5 receptors, or we could achieve the same ends by giving ketamine. And this slide just makes that point again. So in summary, what I've presented to you is the notion that ketamine is the first of a new generation of treatments for depression. It acts rapidly. It's anti-suicidal. It works for people who don't respond to other kinds of treatment. And it may help to promote fear extinction. A single dose, the benefits of a single dose may be extended by repeated dosing. And in the context of repeated dosing, ketamine appears to be safe. And when those repeated doses are administered in the clinic, there does not appear to be much addiction risk. And this perspective, this new neurobiology of PTSD and depression, suggests that there may be other mechanisms for treatment that may come from this line of research to help us both understand the biology of PTSD and depression and new ways to treat these problems. Jeff, at this point, I've concluded my presentation, and I'd like to open it up for discussion.
Wonderful, wonderful. John, thank you for really an outstanding presentation on complicated information that you presented in a really lay-friendly way. So thank you for that and for the work that you've been doing. We've received a number of questions, people just asking, so right now, what if somebody hasn't responded to the usual treatments for depression, can they get treatment with ketamine now? Where does it stand right now today?
Right. Well, that's a great question, Jeff, and I think an important one, because there are places around the country that have clinical programs which are generally open to the public, that are not part of research. So at Yale, for example, we have something called the Interventional Psychiatry Program, and I would be happy, if people want to write to me, I would be happy to connect them to the leaders of that clinical program. There are programs in other hospitals, in many urban centers, and if people have difficulty finding the local clinic that's now prescribing ketamine for treatment-resistant symptoms of depression or PTSD, I also would be happy to connect them with centers in their area.
So from your perspective at this point, for people who have refractory depression, the use of ketamine is, for want of a better phrase, ready for prime time at some of these specialized centers.
Yes. So I would say, but people need to understand it's ready for prime time just because we feel like we have accumulated enough evidence to be able to prescribe ketamine as a treatment. But I think people really need to understand that it's still an off-label treatment. In other words, there is not FDA approval for ketamine as a treatment for depression. That FDA approval will probably first come for S-ketamine, which is being prepared for submission to the FDA, presumably sometime in 2018.
And with the S-ketamine, how will that? Tell us a little bit more about that. How it's given and how often somebody will need to take it as a mode of treatment. Sure.
So S-ketamine can be administered intravenously, like the ketamine that we typically administer. However, Janssen is developing S-ketamine as an intranasal infusion, and it's really important to understand that they have developed a device that makes a very fine spray under high pressure, so it exposes the sinus to the ketamine. That way, when you administer the ketamine, it's absorbed through the sinus rather than you swallowing it. There are a large number of ketamine clinics that are around the country and around the world that are delivering intranasal ketamine via a syringe, or through an eyedropper. The problem with ketamine administered that way is that it's predominantly absorbed by swallowing it through the stomach and intestine. And so very little of that ketamine actually gets into the bloodstream and into the brain. So the Janssen device is not only important because it contains the S-ketamine, it's also important because it is a device that allows that S-ketamine to be absorbed through the nasal sinuses in a very effective way. S-ketamine is typically administered in a dose around 40 milligrams, and usually people start out twice a week. It may be possible ultimately to start it less frequently, but at the moment it's started twice a week, and it's been shown that you can taper that to once a week and then once every other week for the majority of patients. Janssen just published a large study, or they haven't published it, but presented a large study where 75% of patients were able to get to something like once every other week in maintenance treatment. It's possible that S-ketamine will be like the regular ketamine that we administer, and the regular ketamine that we administer can many patients be administered once a month, once every 2 months as well. for many patients.
And will the S-ketamine, people would be able to take that at home on their own? They don't have to go to a clinic for that?
No, they would go to a clinic for that. So they would be able to go to a clinic. They would be able to administer the ketamine through the intranasal way, and then when these side effects had abated, they'd be able to go home or go to work or whatever they wanted to do. The effects of ketamine are very short-lived and very well tolerated.
Okay, so for each of them, you still go to a program to get it in order to ensure the reduced risk of abuse, of addiction, etc., and just to make sure that any side effects that are transient wear off before the person gets to go home.
I think that's a good way to describe it. You know, the side effects of ketamine are a little different than the side effects of other drugs. You may have a brief period where your perception of the environment is a little bit altered. You may feel a little bit unreal, things like that. And so I think it's helpful to have people around you when you're taking the ketamine.
But I want to just restate what you said earlier and have you elaborate on it, which is that the current understanding is that those dissociative symptoms are really more of a side effect. They're not the cause of the therapeutic effect. That there's a therapeutic effect. You describe some of the neurochemistry around that, but sort of going through that dissociative state isn't what makes the person's depression lift so rapidly.
Jeff, that's my belief. I don't think that you need to have the dissociative symptoms in order to get the benefit. And that's why we're testing other drugs that do parts of what ketamine does that may have less propensity for those side effects, to see if there are alternatives. I would say that for most people, once they're in the maintenance phase on ketamine, those side effects, they tend to get a little tolerant to those effects of ketamine and accustomed to them. So it's not that the side effects themselves are particularly difficult, but particularly early in treatment, I think it's often helpful to have someone there to support you while you're going through it the first several times.
You also mentioned that there are other medications that have a ketamine-like effect. Could you say a little bit more about some of those other medications that they're working on that's not yet available?
That's right. So one of the medications is called rapastanil, and rapastanil is a peptide molecule that was derived from an antibody that binds to a glutamate receptor. There is a drug which is owned by Biohaven Pharmaceuticals, another NMDA antagonist, which showed some initial effectiveness for the treatment of depression. It was known earlier as an AstraZeneca drug called 6765. Some people may know it under that name. Lenisamine is another name for it. There's D-cycloserine. There is R-ketamine, which is the mirror image of S-ketamine. People are interested in testing that. And there's a metabolite of ketamine called hydroxynorketamine, which is also receiving intensive study. For each of these drugs, people have mapped their effectiveness to one or another part of the action of ketamine, and we'll see whether or not they're effective treatments or not.
You also brought up the important issue of suicide prevention, and in particular now with some of the tragic suicides of well-known people and with the report of the increasing suicide rate across the population. I'd like you to say a little bit more about the potential for suicide prevention with these treatments.
Yes. So first, Jeff, I share with you just the sense of horror about the increasing reports of teenagers taking their lives, of these public figures like Kate Spade and Anthony Bourdain taking their lives. This is a public health crisis. There's no doubt about that. And I think that ketamine may prove to be helpful because it reduces the amount of time that people are in extremis. You know, people often commit suicide when they hit an extreme state of despair. And ketamine, because its antidepressant effects are so predictable and so effective, seems to be extremely powerful in kind of restoring enough hope that the effectiveness of ketamine in reducing suicide in the clinical trials has been, suicidal ideation in the clinical trials so far has been very striking. I would say that reducing suicidal ideation and urges in a clinical trial and actually preventing incidents of suicide may be two different things, but I think the powerful effect that we've seen with ketamine in the trials so far give us a great deal of hope that it could play a role in preventing suicide.
Obviously, that would be a tremendous advance for society. And you also alluded to the real advance in our field and for patients with depression, other illnesses, where instead of having to go to the hospital because they're at risk of hurting themselves and spend a significant amount of time in the hospital until the suicidal risk lifts, they could have rapid-acting effective medication and then go home, continue treatment on an outpatient basis, and they could go to school the next day or work the next day and resume their normal functioning. I'd like you to comment a little bit about that.
Sure. You know, I'm reminded of the suicide note of Virginia Woolf. And in her suicidal note, she says that she felt another episode coming on and that she was certain that this time she would never get better, and therefore she needed to take her life. When you have a drug like ketamine, which is so predictable in its antidepressant effects and so rapid, it has a way of changing your idea about what it means to be depressed. I've seen this in some of the patients that we treated, which is that before, when they had chronic depression problems or in a relapse of their depression, and the doctor would try a new treatment, and if you looked at the data about what percentage of patients would respond to that new treatment, it may only be 10 or 20 or 30%. If you add a new treatment and you have a 75% chance of getting better in a day, it's a different kind of a situation, one that's much more hopeful. And so I do think that there's the possibility that the difference between this as an adjunctive treatment versus other kinds of prior adjunctive treatments could have an impact. I'm not sure I've answered your question, and if I've missed a part, please repeat that.
No, I think that you did really very clearly. I want to go to a little bit on bipolar disorder and depression in people who have bipolar, which is again a very high risk group for suicide attempts and actual suicides. Tell us a little bit more about the use of ketamine and the other rapid-acting antidepressants in this population.
Yes. I think that this is a particularly hopeful area because the large-scale studies of standard antidepressant medications for bipolar are surprisingly discouraging. Well, maybe not surprisingly discouraging if you've had bipolar disorder and you've been taking antidepressants and you've not been getting better. But the large-scale data are very discouraging about the effectiveness of our usual antidepressants in bipolar disorder. And so we've been pushed as a field to study the second-generation antipsychotics and lamotrigine as a treatment for bipolar depression. Ketamine appears to work very rapidly in bipolar depression. If I refer to the work of Carlos Zarate and his colleagues at the National Institute of Mental Health, that they find as early as 2 or 3 hours after a single dose of ketamine that the people with bipolar disorder are already reporting improvements in their mood whereas the patients with unipolar depression usually take a bit longer to show benefits from the single dose, more like 4 to 6 hours before they start to feel much meaningful benefit. So, and the results so far have been just as positive, just as encouraging for maintenance treatment of bipolar disorder as they have been for unipolar depression. So, I think it may be the case. One of the questions that we don't know is whether there are sort of different types of depression, and obviously unipolar depression may respond well to the SSRIs for many, many people, but there are a lot of people who don't respond, who seem to respond very briskly to medications like ketamine. So it may be there's a whole group out there, bipolar depressed patients, anxious and agitated depressed patients, and certain other groups that may be PTSD, trauma patients with trauma histories, chronic pain that may do particularly well on ketamine.
Is there information yet in terms of particular age groups such as adolescents, younger people, and on the other side, seniors?
Yeah, I'm glad you asked that question. To my knowledge, there's one study underway at Yale. We have had some early encouraging data, and now we're doing a trial to evaluate the effectiveness of ketamine for adolescent depression. Michael Block is the senior investigator on that study. And on the geriatric depression, there's an interesting and complicated story. So Janssen did a large study for geriatric depression. The good news is that ketamine was very safe and well tolerated in the geriatric population, which is not a big surprise because ketamine has been administered to the geriatric population for years as an anesthetic medication and as a treatment for neuropathic pain. However, they were worried about the sensitivity of the geriatric population to ketamine. So they started at a low, probably subtherapeutic dose, and they allowed the clinicians in their study to titrate up the dose of ketamine. But the clinicians in the study largely chose not to titrate up. And so the patients who got ketamine in that study were largely underdosed with the ketamine. And so the study just missed showing a significant result at the final endpoint. But might have been positive if the study had been larger, and certainly we would have expected a more robust result if the patients had been treated with the usual dose of ketamine instead of a lowered dose of ketamine. So that, I would say, a definitive study in the elderly still hasn't been conducted.
Well, John, I could say that your message here is one of true hope for people, in particular people who are experiencing refractory depression where they don't get better, and for their loved ones who care so much about them. Hope for a really significant improvement in this tragic suicide rate that'll really have an impact, and an impact not 5 years or 10 years from now, but more immediate, as well as other future discoveries that'll happen, as you described, that this leads to other branches of research, but there's real help around the corner for this. So I want to thank you for the extraordinary work you've done over the years and continue to do in this area, and for taking the time to share that with us today.
Jeff, thank you so much for those kind words, and let me thank you, because BBRF has been such an important organization supporting some of my work and many, many of my colleagues. It's really one of the most, if not the most important organization in mental health. Thank you.
Well, thank you. It truly is a collaboration between our donors and the scientists, which is why there's been such success. So thank you, John, very, very much. I also want to thank everybody for joining us today. All of the research we fund is made possible through private donations, so please consider making a contribution by visiting our website, bbrfoundation.org, or call us at 800-829-8289. This webinar has been recorded. If you've missed any portion of the presentation or would like to share it with a family member or friend, please visit the Events and Webinars page on our website. Finally, I hope that you'll join us again next month when Dr. Randy Blakely, Executive Director of the Florida Atlantic University's Brain Institute and Professor of Biomedical Science at the Charles E. Schmidt College of Medicine, will present Rare Misspellings of the Genome, Dopamine Mishandling, and ADHD. This webinar will take place on Tuesday, July 10th at 2 p.m. Eastern Time. Thank you again for joining us, and enjoy the rest of your day. Take care.